Hi, everyone. Hope everyone's got a cup of coffee or whatever else you're drinking or doing in another part of the world. So you all have to be muted, unfortunately, because there are so many of you. But please feel free to submit your questions or comments or anything else via the chat option, and I will save it and then post my answers to your questions on the website. So we'll go ahead and get started in about a minute. I'll just wait for everyone to join. I'm going to share my screen with you. Still have people joining. Sorry, give us one sec. I think we still have full waiting.
Dios, todo el mundo tiene activadas las cámaras. Todo el mundo tiene activada la cámara.
Your camera, yeah, you don't have to have your camera on. Sorry, people are still joining.
Hello.
Hi, everyone. We'll go ahead and get started in about a minute, but I'm still admitting everyone, basically.
Okay, thank you very much for putting this.
That's all right. I hope you have fun.
Oh, yes, I'm sure I will.
Okay, sorry, I still need to admit them. Okay. All right, so session one, sorry. Come on, people, get in on time. They're still in the waiting room. Sorry, give me one sec. Going up, going up, going up. Sorry, it's already about 2 past. Okay, I think we're joining.
Estoy en clase. ¿Dónde te vas a hacer?
Okay, I think I'm just gonna have to do it, and I'll start talking, and then I'll just keep admitting people, basically. So session one is all about introduction to software that we'll be using throughout the series, and also looking at differentiating between young and adult cortical bone. So I will, in each session, I will basically show a very short PowerPoint presentation, and these slides will be uploaded onto the website, so you don't have to worry about, you know, making notes or whatever. It's something that you can always go back to later. So let's see if this is working. Sorry. Hang on, is it actually, I don't think it's changing. Hang on, give me one sec. It's a bit slow. There we go. Okay, so okay, so I've asked you to imagine that you're working with already made thin sections. So let's imagine that you're looking at a thin section that's placed basically on a stage of this microscope here, and then the information is basically getting transmitted from a high resolution camera onto the screen. So really what we're interested in is what you're seeing on the screen, right? Because we're thinking about the sorts of data that we could get from a histological thin section. So the software, sorry, for some reason my PowerPoint is really not, it's just going very slowly. Bear with me, it's not changing my slide. Come on. All right, we're gonna have to do it that way, unfortunately, I think. Okay, and then we have more people joining. So there are many different types of image analysis software that you could use to do this. So it's not all about just ImageJ. I suggested that you download and have a look at ImageJ simply because it's you can download it for free. It's user-friendly. So the good thing about it being obviously open access is that we can actually do stuff like this. So, you know, in cases of pandemics, we can actually do some sharing of data and research. And it's actually used in most labs globally. There are a bunch of different types of software that of course you could use to do image analysis. Olympus, for example, which is what we use in the lab at the ANU, they have their own software called CellSense. But again, you'd have to actually pay for that, and it's something that comes with the microscope. Some people actually also use geographic information systems. I've put a reference here that you can have a look at the suggested readings at the end of this PowerPoint as well. So the good thing about ImageJ is, if I can go to my next slide. There we go. All right, we're getting there. So the good thing about ImageJ/Fiji is that it actually comes with a BoneJ plugin. So this was designed by a guy called Michael Doob at Imperial College London. And so it's a bunch of different extras that are specifically designed for the analysis of bone. So not just histological images, but anything else. So if, for example, you're interested in doing anything to do with biomechanical properties of cross sections in long bones, you can essentially do that in ImageJ as well. So what we'll do is take a quick look at that. I will stop the share, and then I will share my desktop. All right. So you would have already. Can you all see my screen? I think it's all good, right? Yep. So you would have downloaded already Fiji/ImageJ. So what you get to see is something like this. It's basically just a long, tab with a bunch of different tools that you see here. And we'll practice this properly as we go through the, excuse me, next couple of weeks. But today I just want to show you the very basic features of this. And so what you would normally do is have your image ready, and then you simply drag it into the empty bar that you see here at the bottom of this ImageJ strip. And what pops up is drag and drop. So you pop that in there. What you need to do is actually calibrate the software as well, i.e. set the scale, which I've already done. So we won't really do it today, but we'll do it, I'll show you how to do it next week. So you can essentially do anything and everything you want with the tools that you see here. And for example, if you wanted to count some of the features that you see in your image, you'd simply click on this option, this tool called multipoint. Sorry. And in this case, we're looking at woven bone, which is what we'll go through in a minute. So if I wanted to count some of the cells, I would be simply clicking on them like this. The great thing about imaging software as well is that I don't actually have to look at it and count myself, because the software counts it for me. For example, another thing you may be interested in is perhaps measuring the length of, let's say, this collagen fiber. So you would pick this straight line tool and then simply select, let's say, this part of the fiber. And then on your keyboard, you press M for measurement, and it will pop up with the data that you just collected. So remember, I'd already preset the scale. So the software is actually telling me, is giving me the actual data. So I'd be interested in this bit here. So I know that this fiber is about 826.697 nanometers. So it's not really all just about that, about the kind of simple measurements from images. You can also actually play with the quality of your images, and you can also alter it if, for example, you're interested in things like densities. Sorry. under image tab, you can adjust your image, so you can play with brightness, with contrast, you can threshold it. So again, thresholding is actually quite useful when you're dealing with tissues of different densities, or if there's a lot of noise in the background of the image, you can simply get rid of some of the information that's unnecessary, so that what you actually want to count, for example, some cells or other structures, they become enhanced against different type of background. The plugin, the BoneJ, sorry, it's waiting room. So the plugin, the BoneJ plugin is under plugins tab, so you can actually go here. You can have a look at things like analyzing skeletons, you know, fractioning, skeletonizing, surface areas. It's very good for 3D analyses of trabecular bone in particular as well. So there's lots of different things we can do with it. So if you have some time next week, do have a look at home, play with it a little bit. For next session, I'll actually give you a couple of images that you can practice as well. But if you don't want to do that next week, we'll actually do it together on the screen. So that's just the introduction to the software. So we'll do this. And now bear with me, I'll go back to my PowerPoint. All right, so it's a bit slow. I don't know, maybe it's because the rest of the video was logged on. Okay, you should now be seeing a picture of cortical bone histology. So I assume most of you have some understanding, obviously, of I guess, of the structure of bone. But when we're talking about bone histology, and in particular, when just referring to these workshops, we'll be looking at cortical bone. And so obviously, it's almost impossible to show you the minute details looking at the whole bone. So what we normally work with, and when it comes to actually quantitative data from bone histology, we look at what we call regions of interest. So in publications, you see them abbreviated to an ROI. And here's just the classic example, a cross section through a long bone. This is a femur. And if you were to take a small section out of the cortical bone, what you see is something like this. And so remember, we're looking at a transverse section. So that's what you see on the screen if you're imaging under the microscope. So it's a horizontal slice through the bone. And for today only, because like I was saying, over the next few weeks, we'll be going through different bits and pieces and parts of the bone. So for today, really, what I think we should be paying attention to are the different types of lamellae. So here, just under the periosteal border, we've got what we call a circumferential lamellae. So they're these kind of long lamellae that just that envelope the entire cross-section of the bone. We also have interstitial lamellae. So they're the ones that you find between the circular structures, what we call osteons. And osteons themselves have what we call concentric lamellae. So osteons are essentially, they're going to become your friends for the next few weeks. And osteon, and in this particular example, I'm actually talking about a secondary osteon. We'll get onto that in a second. They are almost self-sustaining functional units that build your cortical bone. So they're very tightly packed. And as you can see from this picture, through the center of each osteon, we've got a blood vessel, and which basically it helps with the blood supply of the tissue. So an osteon itself is actually pretty important to bone remodeling and modeling, which is what we'll get to next week. Okay, I'm gonna have to be changing slides this way, unfortunately. Okay, so one of the other things we can, and probably something that we should start with anyway, before we get into quantifying histology, is differentiating between what is old and what is young. So some of you who are in more kind of forensic side of things, and those of you who are in archaeology, this is something that can come in really handy at some point, particularly if you're dealing with human remains that are of very much compromised external anatomy. So as you know, in our discipline, we would typically rely on the macroscopic indicators or morphological changes with age to inform more or less what the age of death of the individual is. But what we can actually do is also take a small fragment of bone and figure out whether it came from an old or a young individual. And this is something that actually was firstly commented on by Amprino and Bairatti in the 1930s. then in 1960, Jennifer Jowsey wrote up a paper that talked about age changes in human cortical bone. But it was really Curley who put forward a pretty, I guess what at time became revolutionary paper discussing the fact that you could determine age of death from bone histology without basically, you know, having the gross anatomical indicators. Sorry, this is still not changing properly. So a very simple graph, a diagram that Curley published in the paper was basically something like this. So that's really what I want you to take away from today. And we'll have a go in a second in a couple of examples. Just like a lifespan, your bone also has a life cycle. So when you're a fetus, so just looking at the top here, when you're a fetus and in the basically first couple of years of your lifespan, your bone is very poorly organized. It consists mainly of what we call woven bone, which I'll show you an example of in a second. And you grow really rapidly. So these very first couple of years are pretty much that really quick growth. And then as you're going towards the end of your lifespan, you actually, the type of the bone matrix changes from poorly organized to more mature and basically better organized. And it's basically more competent in terms of its structure and strength. And so the two processes, the key biological, physiological processes that your skeleton experiences, of course, is modeling and remodeling. Modeling is very active in the first 20 years of your life, and it basically changes the size and the shape of your bone. Whereas remodeling, actually, it's also active in the first 20 years of your life, but it's one of those processes that your skeleton experiences for basically the entirety of your lifespan. And so as we're going through the decades, the histological, the actual microscopic expression of your tissue changes. So it goes from pure primary lamellar bone to bone that's filled with, or I should say punctuated with, what we call these secondary osteons. And that is literally the premise of how we determine whether someone is young or old. And so some individuals some researchers have taken this a little bit further to the point where they actually came up with specific equations that predict age of death from bone histology. So they'll literally count these secondary osteons and fragmentary osteons, more on that next week, and then try and provide a specific age number. Now, there's been a lot of criticism of that purely because bone remodeling itself is actually quite a dynamic process. It can be influenced by a bunch of different factors. So it's actually quite difficult to do that properly for any given individual. However, what we can obviously do is, provide a simple, quick assessment of whether something is old or young. Now, this is a really beautiful cover from a book about bone histology and fossil tetrapods. Who does not love, paleontology? And this picture here actually is pretty good. I think it will help you memorize how to distinguish what's young and what's older. So this red secondary osteon in the middle here is essentially that indicator of secondary mature bone growing. But what you've got underneath it are layers of these kind of sheets of primary lamellae. So it's these primary lamellae, if we see them, we know that that tissue is still young and it's still growing. So, sorry, I'm gonna have to go on to the next slide. Okay, so woven bone is the bone that's very poorly organized. Collagen fibers in it are basically all over the place. So this is a very good example of that. You can see that they're going in a bunch of different directions. And this is the type of bone that we'll typically see in a fetus and very young infant. Really, if you're looking at skeletal remains that are older than 2 years old, you don't really see much woven bone at all. You already move into what we call primary lamellar bone. And woven bone you also see at sites of pathologies, particularly fractures, healing sites, and things like that, because again, remember, it's bone that's really rapidly developing and it is producing collagen fibers that are oriented in many different directions. Now, here's an example of an adult that is showing some woven bone histologically. And this is actually an adult patient with osteogenesis imperfecta. And on your left side of the screen, that's the woven bone picture that we were using previously in the demonstration of image J. And you can see these collagen fibers are just going all over the place. And portions of the older lamella, what we call lamella bone, you can see collagen fibers are very nicely and tightly packed in sheets. So this is how you know that this one is actually older and a bit more mature. Okay, sorry, I need to change to the next slide. All right, so it is the lamella bone that I want you to remember for the next few weeks. This is what we're going to be working with, as this is mainly what we get to see in archaeological and I guess forensic specimens. And so lamella bone is then split into primary and secondary bone. Remember, primary is the young, secondary is that more adult type of tissue. So what you would actually see is something like this. These are taken from some of my research in different types of archaeological populations. Oops, sorry, go back. Adult bone, as you can see, is just full of these circular osteonal structures that you can see, to the point where we see several different generations of osteons. More on that next week. But the idea is that you see a bunch of circular structures, basically very tightly packed and almost superimposed on top of each other. Primary bone that basically has secondary osteons, but also a little bit of what we call primary osteons. So you can see there's one here, for example, is just a simple blood vessel. And then here's a proper secondary osteon. But right behind there, you can see these nice flat sheets of lamellae that are still indicating that this is a primary bone. Now, this individual on the left is a teenager, and this individual on the right here is in the 40s. So technically, you should be able to take a very quick look at an example like that and say, this is possibly a young individual. And then, you know, if it's something like this on the right, then it's probably an adult individual. Now, a paper that I was involved in a few years ago, led by Rosie Pitfield, who just finished her PhD at the University of Kent in the UK, she actually studied kind of ontogenetic series of human humeri to check at what point all of these primary blood vessels and that primary kind of bone tissue type, at what point it actually gets replaced by secondary bone. And what she found was that this is approximately around 14 years old. So at that time, you start seeing basically mostly secondary osteons that punctuate primary bone that it's becoming almost completely erased. So if you see a lot of primary bone, very few secondary and primary osteons, you're probably looking at someone who's below 14 years old. Okay, now what we're going to do is have a go at a couple of examples. Now, just something that will help, this picture is from that paper by Kelly in the 60s. Hopefully that helps you visualize, again, everything that I've been talking about. So we've got a secondary osteon here. We've got some primary canals here. You can see that they're much smaller, much more fragile. They actually don't have many concentric lamellae at all. In fact, some primary canals don't have any. But others do, only a few. For example, here you can see they're very nicely surrounding the blood vessel. But really what we've got is just a huge amount of primary bone that's basically going almost through the whole thin section. A very good way to determine whether what type of bone tissue you're looking at is by using what we call polarized light. So I'll show you a couple of examples of that. Polarized light will basically help you look at the orientation of collagen fibers. Here we've got an example of transmitted light. So it's kind of just the natural color of the bone that you're working with. But if you put it under polarized light, you get something like this. So it's actually showing you those sheets, beautiful thin sheets of lamellae that you can almost count. And in fact, some people have done that. And then they'll help you determine how much bone there is, you know, if it is secondary or primary. Okay, so perfect. We've got 7 minutes left. Now imagine that you are working with very small bits of bone, and either you prepared them into histological thin sections, or perhaps you sent it to another lab, and then you're getting some images that show you, you know, just some bone histology, and you need to figure out if it's from a young or an adult individual, bearing in mind that that basically some of it could be simply just all primary, or some of it could be completely secondary tissue. So what sort of assessment would you make if it was a really quick thing? So please type Y for young and O for old using the chat option. Okay, number one. What do we think? All right, we've got some coming in. Lots of people. Let's have a look. Excellent. Young. Good job. All right. Number two. All right. Young or old? All right. Let's have a look. Excellent. Everyone's saying old. Fantastic. See, you're pros at this already. This is amazing. Okay. Number three. Sample number three. Oh, yeah, we've already done that one, haven't we? No, hang on. Yes, that's right. We've done that one. All right. Young or old? Excellent. Excellent. No, this is so slow. Sorry. So annoying. All right. How about this one? What do we think? Excellent. Okay, good. All right. And then I think I've got one more. Last but not least. Ta-da. This one's very, very young. Very young. Excellent. Excellent. Okay. All right. Sorry, I'm trying to power. This is so slow. There we go. All right. So basically, in this 25 6-minute session, you've just learned how to determine whether a bone sample came from a younger or an older individual. You should also have a very basic understanding of how to operate image J. And next week, we'll move into those secondary osteon densities and their relationship to remodeling. I'll talk a little bit about the process of that, and then we'll actually have a go at estimating osteon population densities. I'll also talk you through the different types of secondary osteons that we see, that we can see in bone, and simply, you know, how we determine fragmentary and intact osteons. And that's basically it, folks. And here are just some references that you can follow up later. But other than that, that's it for today, and well done. I'll see you next week. All right,
Thank you.
I think I can unmute you. We still have 3 minutes if you want to ask. We probably don't have too much time, but the idea is to keep these things just very short and sweet and to the point. So you can take away one little bit of information or some knowledge, you know, each session.
How will we access the PowerPoint?
I will upload everything on the website, and you get a link. You'll get a link to the next session in my email. I'll do it basically now. And yeah, so just keep checking your emails, and if they go to your spam, please always check your spam or junk folders, but I'll be sending information via email. So I'll send a link to the next session right now, and yeah, I'll upload everything on the website.
Thank you very much.
Is there any way that we can determine the exact age of the individual that we're working with?
You can. Are you asking whether we can do it? Sorry, can you repeat that?
I wanted to know if it is possible for us to estimate the exact age of the individual that we're working on.
So I mentioned that briefly. Some people have attempted to do that. It's still a very gray area. Some researchers have supposedly shown that it works, so they correlated chronological age with histological age, but then a bunch of other researchers have shown that it doesn't work because bone remodeling responds to more than just age, basically. And so it's a bit of a gray area. If you're interested, I can follow up with a couple of other references and you can read up about that. I am very skeptical about doing a specific estimate just from osteonostructures. So I would just follow, you know, that idea of you can tell whether bone matrix is young or old. But yeah, I will leave it at that.
Thank you.
That's okay. Okay, enjoy the rest of your day, or have a good night, everyone. I'll see you next week. Bye.
Thank you.
Bye. Thank you.
Thank you.
Bye.
Thank you.
Thank you. Bye bye.
Bye.